Here are the answers to the critical thinking practice questions for Chapter 8. So in this first one, I asked you to figure out what amino acid sequence would be formed from this DNA sequence or template strand. So the first thing that I would do if I were answering this question would be to divide these nucleotides up into threes. So I'm just going to zip right through there and separate them into threes. That helps me keep track much easier. Now, before I can go to amino acids, I have to go to RNA. I have to make an mRNA from this DNA transcript. So this is the mRNA that I would get. So A goes with T, U goes with A because there are no T's in mRNA, G goes with C, A goes with T, G goes with C, U goes with A, G goes with C, T goes with A, G goes with C, G goes with C, U with A, because remember there's no T in RNA, A goes with T, U goes with A, G goes with C, A goes with T. So now what I do is, now that I have the mRNA, I take these codons and I figure out from the codon chart which amino acids these code for. So to find AUG, I go down to the A's and there it is right there. So MET or start is my first amino acid. All proteins start with MET. We may clip methionine off later, but they always start with methionine. Now I need to find AGU. So AG's over here. AGU, that's serine. CAG, here's the C's. Here's the A's. There it is, CAG. That would be glycine. GUA. Let's go down here to the G's. Go over here. Oh, here's the U's. GUA. That would be valine. And then UGA. I just happen to know that's one of the stop. And so that would be the answer to that question. So next, I ask you some mutation questions. So I still have the same template, but I'm talking about changing the DNA. So in the first one, nucleotide 6 is changed to G. What type of mutation is it? So I count over 1, 2, 3, 4, 5, 6. So this one got changed to G. Now, before I can figure out what kind of mutation it is, I need to figure out whether there's been a change in the amino acids or not. So I bring up my mRNAs. Now I've got to change this U to a C. And here's what I had before. So if I look for A, G, C, so here's my A's, here's my G's, there's my C. Oh, so I still have a serine. There has been a change in the nucleotide sequence, so it's definitely a mutation. But there hasn't been a change in the amino acid sequence, so it's a silent mutation or no change in the amino acid. Now, let's take a look at this next scenario. So, if I change nucleotide 5 to a T, what type of mutation is it? So I'm going to count over 1, 2, 3, 4, 5. So I'm going to change this C to a T. So bringing back my mRNA. So I've got to change this G to an A. So I'm looking for AAU. So the A's are down here. AAU. Oh, I've changed it to this ASN. There's been a change in the amino acid sequence. So what kind of mutation is it when we've definitely had a mutation. We've had a change in our DNA sequence, and we've had a change in the amino acid sequence. That is a missense mutation. Now, just for the fun of it, what if I'd changed one of these over here and I had a stop here instead? What kind of mutation would that be? That's right, it would be a nonsense mutation. Alright, on to the next question. You expose a lab culture of bacteria to an antibiotic. The antibiotic kills the bacteria. Analysis shows normal levels of DNA and RNA, but reduced levels of protein. What is the cellular target of the antibiotic? Now you may be thinking, wait, we haven't talked about antibiotics. Well, you can still answer it from what we've discussed in this chapter. So let's take a look at our answer options. Are we attacking DNA polymerase? Well, let's look up here. If I attacked DNA polymerase, what would I stop producing? Well, I would stop producing DNA. Well, I've got normal levels of DNA, so that can't be the right answer. Protein structure. Well, if I had reduced levels of protein, maybe the antibiotic is attacking the protein structure. Let's keep that one in mind. Ribosomes. What do ribosomes do? They use the RNA transcripts of DNA to make protein. So that's definitely a possibility. Now, what if I attacked RNA polymerase? Well, what do RNA polymerases do? Well, they make RNA. And if I didn't have any RNA, I wouldn't have any proteins. So I would have reduced levels of RNA and protein. So, nope, that's not the right answer. Enzyme function, that's definitely a possibility, but it's too broad. DNA polymerase, RNA polymerase, ribosomes are all enzymes, but we've already shown that it can't be DNA and RNA polymerase, so this is too broad. So it's either got to be protein structure or ribosomes. I'm going to go with ribosomes because that would show reduced levels of proteins. Whereas if the antibiotic was attacking protein structure, then I would be attacking all of the proteins in the cell, including DNA polymerase, ribosomes, RNA polymerase, and enzyme function. So protein structure is too broad. Now for this next question, we're going to be talking about horizontal gene transfer. So here's our scenario. Suppose you inoculate 3 flasks with Staphylococcus epidermidis. Flask A contains 1 phage, or bacterial virus, if you remember to Chapter 5, that contains the gene for resistance to penicillin. Flask B contains several copies of naked DNA containing a gene for resistance to penicillin. Flask C contains just media. After a few days of incubation, you add penicillin to all 3 flasks. Which flask do you predict will have live bacteria after 12 hours? Well, I'm going to eliminate flask C because it has just the media and the S. epidermidis. There's nothing in there to help our S. epidermidis be protected. So anything that has C in it, I'm going to eliminate that one. So my question is it A, B, or A and B? Well, in flask A, we've got transduction going on where we have a virus, or a phage, a bacterial virus, that brings the gene for resistance to the S. epi. That's all fine and good. That would protect it. And as the cell divided, that gene for resistance would be in each of the daughter cells. So through vertical gene transmission, resistance would be passed on to the rest of the cells. Now, here in flask B, we're talking about transformation. Naked DNA being picked up by the Staphylococcus epidermidis. So once again, each of the cells that manage to pick up the copy of the gene for resistance to penicillin, when it divides, it's going to produce daughter cells that are also resistant. So the answer is D. It's A and B. Now for the next question is, which will have the highest number of live bacteria? So which one's going to be more turbid? Well, let's back up real quick. So is it going to be from flask A that had 1 phage that had transduction? Okay, so the phage is targeted toward S. epidermidis because that's what viruses do. Whereas in flask B, we've got several copies of naked DNA. The bacteria have to run into the DNA to have transformation take place. So which one is going to result in more cells having resistance? So which one's going to be more turbid? Well, even though transduction is targeted, we're still talking 1 copy of the gene versus several copies. You have a higher chance of cells running into the naked DNA. So I'm going to go with B, the flask that had more resistant genes available to the cells initially. Alright, on to this next one. You isolate a strain of bacteria that divides much slower than others of that species of bacteria. Analysis shows a mutation in the DNA polymerase. Okay, so we have a DNA polymerase mutation. Which of the following could explain the slower division time? Well, I've got two answer options that talk about RNA. DNA polymerase does not make RNA polymers, so I can eliminate A and E right off. So I'm left with B, C, and D. DNA is replicated slower, but more accurately. Well, that would certainly explain why this strain of bacteria divides much more slowly than others of that species. It makes sense. If you have a mutation in the DNA polymerase that's proof-checking more thoroughly, it could slow things down. DNA is replicated at the same speed as before, but with more mutations. Well, this doesn't talk about slowing down the division of the cells. So it's nutsy. DNA is repaired with less efficiency, leading to more mutations. Well, we did have a mutation, but it doesn't talk about this strain of bacteria having more mutations. It just talks about it dividing more slowly. So the answer is B. Okay, last question. You are studying lactose metabolism in E. coli. That means you're studying the lac operon. Remember, that's an inducible operon. Which of the following would be a good way or ways to study how lactase production is switched on and off? Because remember, in critical thinking practice, sometimes I have more than one correct answer. So let's take a look first at the question. So we're talking about regulation of gene expression. So what are we talking about? Transcription. Okay, so we can eliminate any answer option that does not deal with transcription. Grow the E. coli without lactose. Okay, that's a good start. Then incubate the E. coli with labeled DNA and RNA nucleotides and lactose. Okay, now first question is, why would we label the DNA and the RNA and not the lactose? Well, the lactose turns on the lac operon. DNA and RNA labeled nucleotides would show us that we were getting polymers, new polymers of DNA and new polymers of RNA. When we're looking at just transcription, are we looking at new DNA polymers? The answer is no. So we can eliminate this one. Expose the E. coli to mutagens to produce knockout mutants of the lactase gene. Well, that would certainly help us understand the structure of the lactase gene and what parts are important in making the lactase. But does it talk about switching on and off the lactase gene? Okay, answer is no. Grow the E. coli without lactose. That sounds good. Then incubate the E. coli with labeled RNA nucleotides and lactose. Okay, so we've got the lactose to turn on lactase production, labeled RNA nucleotides to look for new mRNA transcripts. That looks like a good answer. But let's look at this last one. Compare the genomes of E. coli that have been isolated from humans with and without lactose intolerance. Well, that would be an interesting study, but in this answer option, we're really asking the question about the genes for lactase in humans with and without lactose intolerance. We're really not looking at the genomes of the E. coli in these different types of humans. Probably whether humans produce or don't produce lactase would not affect the genomes of the E. coli. I could be wrong. Basically, this answer option does not address turning on and off gene transcription. It's looking at genomes, it's looking at the DNA, it's not looking at turning the genes on and off. So, C is the best answer.